same day as the soccer game?
No. That's why I needed to move the class up. Because
Any class at all?
I don't know. So they didn't tell us what to do. They didn't tell us when you guys were going to be going to soccer or how long. They have given us very little information about a lot of things. So what?
The syllabi of them say we have this class.
Well, again, but if you look at my syllabus, it just says soccer. If you look at we'll go to my web. Yes. So I'll show you guys where to look for things. On my, because it says, because it's Wednesday and we could have it in the morning, but I didn't know how long it was going to take you to get there. Again, I had no idea how far away the soccer stadium was. I had no idea when the match started. They just said, do you want a ticket? And there's 20 tickets, and that's as much as I knew. So I didn't know if I could have, and I didn't want to schedule class that was going to interfere with people who were going. How many people got one of the free tickets? Oh, nice. So, oh, yeah, that's it. So, and also if people were going to be gone in the soccer game, I didn't want to disenfranchise the people who were going to the soccer game by having class where they couldn't sit in. This is why I moved everything up a little bit so we could start on chapter 4, look over chapter 4. We're going to go through chapter 4 all total. Whoops. Bonjour. Bonjour. Oh, Gabby. So you guys, I wanted to show you, somebody was asking about our schedule. And so always look at the lecture information schedule. That is the most current one and the most accurate one. You'll notice that the lecture information one is now already slightly different than the one in the syllabus. But that's because I had to give something, and I didn't know anything about this. I mean, this is my first time teaching abroad. This is my first time being here. So I didn't know anything about like how long soccer was going to take or anything like that. So here you can see today we're here. We're gonna, today we're here, right?
Yes.
Yes, so this is the chapter 2. We're gonna finish chapter 2 today. And then Thursday we're gonna start on antigen capture and presentation. Now the other thing is it might be that we run over and we do a little bit, we still have to finish a little bit of innate on Thursday. So this is a general idea. You should read chapter 3 before Thursday though, okay? Because who knows? I don't know about pop quizzes, but there could be a pop quiz of some sort, okay? Of some sort, yes, where you have to draw something, like maybe a lymph node. Hint. On Thursday, or so again, I'm also going to ask you guys, there might be a very basic question over something in chapter 3 that you should know just from reading chapter 3. Not a small little detail or word, but just a general thing, so that you guys all read chapter 3, okay? And I want all of the groups to be together, and I want us all working together on this, because really, we are down 1 week already, right? And so it's just going so fast. So, and it's so exciting. Then we have Friday where we're not even here. So this is the most accurate. Somebody was asking about soccer. Because I didn't know when soccer started or where it was or where students were going to have to queue up or how long they were going to be gone. We don't have class on Wednesday. So anybody who didn't get to go to soccer, get your toes done or go downtown and buy something fun. They can go off and watch soccer. So we don't have class on soccer. So, but we are going to review all of chapter 4 on Tuesday so that when we come back, it will be on the exam. So chapter 4 is going to be on the exam, and chapter 4 is hard. So it also gives you Wednesday to review chapter 4. And then when you come back, we're going to ask questions on chapter 4 where you're like, I don't get this. Go over this again, okay? So that's what we're going to do. Chapter 1, 2, and 3 are pretty straightforward, okay? Chapter 1 is basically the new materials lymphatic system. Everything else in chapter 1 is going to be kind of like, we're going to review in more depth and review in more depth. In the medical school, we call this cycles. So you're going to go through cycles where you start here and then you build, and then you start here and you build, and then you start here and you build. Okay. So this is the next 2 weeks through June 14th. Okay. So consider your book, your notes. I'm not going to take anything from, it might be a thought question, like what would happen if something was missing? What would happen if something didn't work or was genetically altered so that you couldn't make a certain complement molecule that we're going to talk about today? So I want you to think about, those could be questions. It's not just going to be memorization. Fill in a blank. What is this word? I do kind of want you to think, and I want you to start asking questions. The other thing is, while we're getting started, I want you to do a Google search on NK cell expansion, clonal expansion. Just do a Google search. And you don't have to, and Haley, if you don't have a computer, look at somebody else as well. They're doing a Google search. Or look for any, you can talk about any granulocyte. You can look for mast cells. What's going on with mast cells? Do a couple of searches with mast cells or PMNs. We're learning all sorts of things about the innate immune response that we didn't think was true. So what's still true about chapter 2 is that those receptors are still restricted. You can think of them as the dumb version of, oh, this stupid thing keeps wanting to install PowerPoint on my computer every time I turn it on. So I'll get that fixed. Let's do this. Whoops. Nope. Let's go back. Let's go back and do this. Okay, and so how many of you remember anything from yesterday? Because your brains are still asleep. Yeah. Okay, so we're getting there. So again, what is always true is the receptors on the innate immune response are less diverse. And what the book did say really well was that there are only 2 receptors for the acquired immune response, B cell receptors and T cell receptors. But we have millions, trillions of flavors of each one of them, okay? Here, they said about 1,000. There are 1,000 set receptors. So TLR4 will always recognize LPS. So those receptors will always recognize what they're supposed to and so any bacteria that has LPS, and for those of you who haven't had micro, Shigella, Salmonella, E. coli, Vibrio cholera, all of them are gram-negative. All of them have different forms, different shapes, different virulence factors. All of them are gram-negative. All of them have one commonality. They all have LPS. Okay, they're gram-negative. So again, it's one thing that we can identify with any time. So the innate immune response is like, boom, gram-negative. Oh, gram-negative, something's happening up there. We seem to get an overwhelming response of dendritic cells going up on dendritic cells. And they talked about the cells that, the sentinel cells are the ones especially that display these, because these are the ones that are going to be manipulating the immune response. So depending on which set of toll-like receptors are pressed on a dendritic cell, it's going to migrate into the paracortex, and it's going to tell the T helper cells there to start mounting an immune response to what it saw. Now they can't see, the only way that they know what's there is to have these receptors trigger, right? Does that make sense? Okay, so, and again, when they say that host cells aren't acted against, this is mostly true with the innate immune response. We have very few autoimmune diseases where the innate immune responses are involved. However, the biggest problem that we do have is when the innate immune responses are compromised in some way. And so we'll talk about that in a little bit, okay? So we talked about these, we talked about them being standard again. LPS, everything has LPS. All of those are gram-negative. They all have 4 wheels. These are some of the things. Again, we have obligate intracellular viruses. And when we talk about an antibody response, once this virus is invited into the cell, once it gets in and uncoats, that viral genome simply boards the cell. And back in the olden days, there was something called Star Trek. They used to have something called the Borg. And the Borg were things that would take over and force you to conform to their way. I like to also think of them as vampires. If you invite a vampire in and he bites you, what do you become? A vampire. Same thing for virus. If this virus gets into the cell is no longer a liver cell. It's no longer a muscle cell. It is no longer a functional cell unless we can fight it off. But most of the time, we simply have to kill the cell because the virus is actually too capable of overwhelming each cell. Once the virus gets in, it is going to cause hepatitis in liver cells. It's going to make more, and all it wants to do is make more viruses. It is a genetic program to make more viruses. That's all that's in that DNA. The other thing that you guys don't need to know is every virus is extremely complex on how it creates its variants. You guys don't have to know each one. Some of them are so complicated, I don't understand how they get made. And I've gone through it 3 or 4 times. I still don't understand coronaviruses in some ways. They're extremely complicated. And it's like a computer program that gets turned on genetically. Once it's like a computer virus. Once it's downloaded into your computer, that virus unloads its genetic code and overwhelms the normal system, and all you can do is what it wants you to. It's going to make viral proteins, it's going to make new viral genomes, and when we're making new viral genomes, it's generally when we're making the double-stranded RNAs, if it's an RNA virus. Viruses come in 2 forms, DNA or RNA. DNA or RNA. They are never both. DNA, RNA. And the actual people who study these viruses, they go to meetings, DNA viruses. They do not even talk to each other because these viruses are such different kinds of viruses. RNA virologists go off to RNA meetings. DNA viruses go off to DNA meetings because they're so different. Herpes viruses are DNA. Influenza or RNA. You guys don't need to know those, but I mean, so herpes virologists go off, they study chickenpox, they study herpes, they study cold sores, they study all those things, Kaposi sarcoma, all of those things. So these are what viruses are. They have viral spikes, and every viral spike is different. Every virus causes a different type of infection, a different type of infectious process. So the immune response has to respond differently every time we get a new virus. Every time we see something, we're going to respond. And the other thing that's weird about this is that the virus fights back. How does it know how to fight back? There's a virus, and we're going to talk about how they try to inhibit the immune response. HIV is really, really good. Has anybody had a couple of lectures on HIV or know anything about HIV? You know, tell how complicated it is, right?
Isn't it because like it infects CD4 plus T cell?
It infects CD4 positive, but the other problem with it is it has probably like 12 accessory proteins made. And every protein has a way of sticking a spoke in the wheel of the immune response. NEF is going to do this, so you can't do that. VIV is going to do that. These different, every single one does a different process. And we keep finding more and more ways that these little proteins that are made, not even as the main 3, not even as protease or reverse transcriptase or integrase, not the main 3. These are the small little accessory proteins that keep getting involved in the immune response and stopping it, which is why we have such a problem with HIV. They are extremely, extremely complicated. Okay, so we're going to talk about all of these. I did miss one. We're going to talk about C-type lectin receptors, but most of the time these are all just static receptors that are on the surface of every, there are CD names for each of those C-type lectin receptors. There are the TLRs, I think, are named TLRs. Again, other receptors, we're going to talk about these things. And the book, I noticed, did talk about CPG, non-methylated CPG, as a motif that's found in bacteria. The interesting thing about viruses is that viruses use whose replication machinery? Ours. So most of the time, viral, the double-stranded, if it makes DNA, it's going to make DNA the way that we make DNA. So it's going to look a lot like our DNA, right? Because it's using our methylation pattern, our DNA replication machinery. One of the things that we can tell that's different between their RNA and our RNA is that our RNA has to be methylated or capped, or capped at one end on the 5' end, and then it's polyadenylated after it's made. So if it's not capped or polyadenylated, we can also say something is wrong with that. And there are TLRs in the endosome that recognize that, okay? There are Rig-like receptors, RLRs, that can recognize. The primary activator of RLRs are double-stranded RNA. And we actually use that in a lab situation. If we want to activate cells, we actually just have double-stranded RNA and we pop it into the mixture and the cells just go boom, right? Because all of a sudden there's double-stranded RNA everywhere. So, you guys do not have to know the names of each of the TLRs with the exception of TLR4. You know what? I'm going to make a change to that. I'm going to say you need TLR4 because it's a surface TLR, and then we're going to pick one of these that you need to know for an endosomal. Somebody pick one. TLR3. Okay, TLR3 and 4. Okay, so TLR3 recognizes double-stranded RNA. Okay, and it's in the endosome because that makes sense, because that's where we would find a virus. We'd have to digest that viral coat away to find double-stranded RNA, right? Or the other place that this could happen, and I want you to think of this, is when we talk about phagocytes, and we're going to talk about phagocytes today. A phagocytic cell is going to eat a dead cell that had been infected with viruses. So inside this dead cell are a lot of viruses. So when it eats, it gets into that endosome. There are double-stranded RNA inside the cell. We now have this cell in here, right, full of viruses. In the endosome, once we digest that, once we destroy that, and that's what the endosome is going to do, we can find those breakdown products. We can find those viral particles that could also indicate what's going on. So this is one of the ways of getting something that's inside a cell into, or outside of a cell, into a different pathway that we're going to talk about. I just want you to think about that now, okay? Does that make sense? Do you have a question?
Yeah, so it senses it and then it creates a signaling pathway that starts the immune response. That's what you're saying. Okay.
Exactly. So the thing is we have TLR4 is on the surface for interstitial extracellular bacterial infections, primarily. Because if you look at what they're showing up there on the top, because viruses don't exist for long outside of a cell. They have to get inside of a cell. That's where they want to be. So everything up there is bacterial, 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 bacterial. So the surface TLRs are recognizing bacterial, extra, outside the cell. These are what's inside the viruses or inside the cells that you're going, because we're digesting them, okay? Or we're going to break down the bacterial products. So we're going to take, if we phagocytose a bacterium, we're going to throw toxic molecules that we're going to talk about in just a minute at them, and they're going to break them down, and then we can recognize that there's products there, that these molecules are sent. And then these are those signaling, those tiers. These are those tier signaling. If this gets sent, it sends a specific signal that's different from this. Bong, bong, bong. Again, it's the combination of all the ones that get pressed at once that tells this cell what's happening. That's how it knows what's going on in the world around it, in its environment, right? So these combinations are all going to send a signal, and depending on the signal sent, is it going to cause acute inflammation? No matter what, it will always stimulate the acquired immune response, okay? So NF-kappa B, always triggered, always triggered. We are going to see some of the differences depending on what gets triggered and how. If this gets triggered, it's going to be more along the lines of IRF responses. But as an immunologist, I can tell you we always track IRF responses in almost every infection. Because we look at these are common transcription factors that act on aspects of an immune response regardless. So these are two very important transcription factor families. NF-kappa B, and we're going to talk about this again much more in detail. The other thing the book wanted to tell you about, you guys don't really have to know, well, again, it talks about NOD1, NOD2, and NLRP3. NOD1 and NOD2 recognize something called peptidoglycan, which is a component of all bacterial cell walls. So, except for one. So there's an exception to every rule. There's one bacteria that doesn't have a cell wall, but every other bacteria almost has a little peptidoglycan in it. So it makes sense that if an intracellular bacterium gets in, it's going to have peptidoglycan in it, and NOD1 and NOD2 are going to recognize it. So again, innate, standard, there's going to be peptidoglycan here. If there's peptidoglycan, we have alarms that set off peptidoglycan alarms, right? So that's what NOD1 and NOD2. NLRP3 can actually recognize a whole bunch of products, and even viral DNA, right? So it not only recognizes peptidoglycan, but it recognizes a whole bunch of things. And you can think of this as being a set of, you have to think of this as being a set of like Legos. They have to, once those things are detected, once something comes in, those three different molecules come together to form this inflammasome. And only NLRP3 creates this inflammasome type situation. And what do you think inflammasomes are going to do? Cause inflammation. Jeez. Yes, okay. We're going to cause inflammation. So we have the NLRP3. You need to know that the sensor NLRP3, there's an adapter and then inactive caspase-1. And the only reason they talk about this is one of the primary, because this is such an important pathway. You pharmacists, when we take an analgesic, something that stops inflammation, stops a fever, you're going to try and figure out how to break down this pathway, right? So you're going to learn all the signaling pathway I'm going to show you is a tiny little part of the signaling pathways you're learning, right? Are you learning? Oh God, yes. So it's worse for them, just so you know. So we're going to activate this. Once this is activated, caspase becomes activated. So it goes from its inactive form to its active form. So we have NLRP3, an adapter inactive caspase-1, come together once it senses bacterial products. We're going to then activate this caspase-1, okay? And then innate signals TLR, we'll just say NF-kappa B, one of the transcription factors. So these are the TLRs have sent a signal into the body saying bacterial products are here, especially LPS is very much a pyrogen. Think of something that causes heat fever, right? So, and anything with LPS in it is going to cause a fever. This is one of the trademarks of that. Where also some viruses, if you have, or some bacteria, if you have a fever in summer, boom, you know that there's something wrong. It's pyrogens are going on. Okay, so here we have the innate signal says, we're making an inactive bomb, okay? We're gonna make it inactively just in case we're wrong. Because we don't want to having a fever, having inflammation is bad for us. It causes the activation of all sorts of toxic products that can be as harmful to us as it is to the bacteria that we're trying to fight off. So we're gonna make this in an inactive form, and this is one of the cytokines that the immune system is going to make to warn what's happening, to cause inflammation. So you guys need to know something called IL. IL stands for, let's write it down. Let's see if I can write now. Oh, go back. What do you think interleukin stands for? Does anybody know what a leukin is, or leukocyte? White blood cell. So, interleukin, what do you think talks between? And besides what, it's the way leukocytes are talking with each other. So, we're going to talk in class about, this. This is now different interleukins. And I want everybody born in February now. No, let's make it January. Everybody born in January to raise your hand. Okay? You've upregulated the receptors. Upregulate the receptors. Okay, this is going to be mean. Can they bind to that? Could they bind to that? Oh. Okay, that was a better tosser. Okay, let's try it again. So now, I'm going to do it again. I'm sending this out into the bloodstream. I'm sending out IL-1 or another interleukin. It's mine. It's a problem that would be better with a ball. I was going to buy balls, and I'm like, no. Because then I have to take them back with me. And you're like, what are you doing with these balls? And you wouldn't understand. I can talk to a specific set of cells depending on the receptors. The January receptors were upregulated. None of you else should be responding because it's not your job. Only the cells that can respond. So I can send information long distances with cytokines, with interleukins. However, we're going to talk about, there are some, when we talk about cell-mediated responses, that my receptor has to bind to your receptor. And if that binding is blocked, you will not be activated. Okay? However, if you have a receptor and you don't, I can activate this one and not this one because the cytokine, the transcription factors that were triggered that activated her were not activated in you. She's the chosen one. She is now Neo. I don't know, have a movie. Maybe we'll have a movie marathon. We'll either watch Contagion on Wednesday or The Matrix. Oh, so if you've seen the movie Contagion, just so you know, as immunologists, they were consulted by the CDC and the World Health Organization to make something as accurate as possible about what would happen in case of a pandemic outbreak. That's what it's not if it's going to happen, it's when it's going to happen. This is what most of us believe. So, really, truly horrifying, isn't it?
Build my bunker.
Yeah, exactly. Well, this is why people go to Montana and just live off the grid. Because they don't want, those will be the people who may survive. So, yeah, no.
They won't even realize that the world has been infected.
What?
They won't even realize the rest of the world has been infected.
True. Well, most of them have internet because most of them are online geeks and they're playing some World of Warcraft with me. Yes, okay. Okay, so let's go back. And I'm on the wrong slide. So we're going to have an inactive form, pro. So anytime you see the word pro, it's not the better form, it's the inactive form. So here we have pro IL-1B. We have it secreted in an inactive form. The only way that we can get it activated is have the activated caspase-1 bind to the pro IL-1B, interleukin 1B, and then it gets secreted and causes inflammation, causes a fever, causes all the things that inflammation, we're going to learn inflammation does. Okay? So, this is pro IL-1B. And the, I beg your pardon? Yes, Anna.
What activates the transcription of the pro IL-1B?
Toll-like receptors being bound to microbial products.
Is that different than, so that's like a different sensor than
the NLRP3 that activates the caspase line?
So again, so that's a great question. So you're worried about things outside or inside working together, or how this gets inside to cause these things. The body can sense, we can sense bacterial products on the outside of the cell. So some of these bacterial products can come in to cause the activation of NLRP3. So it doesn't have to be, it could be an extracellular bacteria that also triggers the activation. There's different ways that these products can get inside the cell. Okay, so again, that's a great question. You just have to assume it works, okay? Because the way it gets in is above your pay scale. And again, you guys are learning this much of immunology, and there's this much that I don't even know about, right? So I go to graduate theses presentations, and they're talking about one aspect of one interaction of one of these molecules with 87 different other 3-letter acronym names, and any one that gets taken out, this other one's going to come in, and we figured this is our smallest byte, un petit byte, okay? So this is our smallest information that you need to know to understand basically what's going on, so that you can kind of Dick and Jane run. This is Dick and Jane run immunology, okay? So, now we're going to talk about the skin, and this is what gets my goat. This is what bursts. I hate how they just simplify the innate immune response and how important the skin is and your physical barrier is to your health, and how important. Not only that, the other thing is it's a physical barrier to infection, but the other thing is they're not even talking about is the need for normal commensal microbiota on the surface of your skin. If you don't have normal commensal microbiota, it allows things that cause disease to colonize and infect. We have good bacteria. Like they were talking about the fort that was built around the wall. You could think of that fort is our body. The skin is the wall. And outside the wall were all of these little people who came, farmers and stuff, that supported the people on the inside. Because they lived on 9 hectares, so you can't feed 10,000 people on 9 hectares. So you need farmers on the outside. You need normal people on the outside, things that are working and allowing you to live. So you need to put normal commensal organisms on the outside. So we have normal commensal organisms all over our body. And the other thing that's really weird and wonderful is the commensal organisms on your forearm are as different from the commensal organisms on your forehead as the desert from a rainforest. And you can think of it, think about the oil on your forehead. Think about how dry and salty it is on your forearm. The organisms that live there because they have found a niche they can grow in and on, right? So that's why microbiota are just as important. The other thing that we did not realize is that for a long time we found antibiotics, and the only place that we find antibiotics is we stole them from bacteria and molds. Penicillin comes from a mold. We are not nearly smart enough to make penicillin. I don't even know that we would ever have come up with it until Fleming happened to notice he was crap at isolating. He left his plate out too long. It got infected with mold, and he realized that the mold was inhibiting the growth of his Staph aureus. And he's like, I would have thrown it out. He's like, hey, look, I wonder if that's secreting something that's acting on the bacteria. And then he abandoned his work because he couldn't figure out how to isolate it. It's too complicated. There's Ernst and Chain, who won the Nobel Prize. They gave it to Fleming too for finding the observation. But for isolating penicillin, we're too dumb to make it. And we find from molds and bacteria in the soil, normally most of the antibiotics we get are not found in or on the organisms that grow in humans. They're soil organisms that we've happened to isolate and been able to steal their ability to have microbial warfare. This is true, microbial warfare. They're going to try and kill off bacteria that are trying to invade their space. No, this is my space, you can't live here. So they secrete antibiotics into the soil so other bacteria can't live there. So we found that we make peptide antibiotics that are very similar to these things on our skin and in the crypts of our small intestine, where those crypts were on where the lymphocyte, the Peyer's patches were. At the base of those crypts, we make a lot of these peptide antibiotics, defensins and cathelicidins. They're in your book. They're in your notes. They're already there. So defensins and cathelicidins are the human antibiotics we use to stop bacteria, to try and people say, well, how do normal commensals live in the presence of human antibiotics? They've learned to evolve to live there. They are the ones that have figured out how to survive on us in that spot. Does that make sense? So you just have to accept that it happens. Question?
What was the name of the types of antibiotics?
It's in your book.
Okay.
Let me just show you. So I'm going to show you a couple things at our break. We're going to take a break at 10:30. Okay? No, that's great. And I'm not going to write everything that we talk about that's in the notes in the books on the slides, because I do want you to use your book as your notes. And they talk about this exactly like that. They call them defensins and cathelicidins. Okay? And those are the two forms of peptide antibiotics that we make that are very similar to other types of antibiotics. Okay? And then we have something called these, we're going to call them IELs, for intraepithelial lymphocytes. So we have IELs, and again, I will write this down because it's not clear. We call them a gamma delta T cell. And the reason we call them a gamma delta T cell is because these gamma delta T cells are a dumb form of T cells. They have a very restricted T cell receptor. So instead of going through positive and negative selection where you're elite fighting troops, these are the people who stand at the front line and they just go, oh, and that's all they do, right? They are little tiny ways to try to kill things if we need them killed quickly. And the other thing that's different between these is we're going to talk about the difference between these is that our cells of the acquired immune response, our T cells of the acquired immune response, are called alpha beta T cells. And it's all about the T cell chains. Okay, and we're going to learn more about this in just a little bit. Okay, so the T cell receptor on these dumb ones have a gamma delta T cell receptor. The T cell receptors on our smart ones have an alpha beta T cell receptor. We'll learn more about this in just a little bit. Okay, but these are dumb T cells. They don't recognize the same thing that our acquired immune response recognizes. They actually recognize something called lipids. So it's one of the ways that we can identify the body stops bacterial infections in a different way. That's as much as you need to know about the gamma delta. So I just ripped this off from my book. So those of you who have had micro before, I wanted to talk about the skin in more depth because it's so important. It is so important. So when we have the skin, our body is covered in two forms of epithelium. We either have mucosal epithelium, and you guys remember from that Peyer's patch that the mucosal epithelium is one layer of skin, one layer of cells thick. Okay? The way that we enhance that defense mechanism is we have these cells, they're called goblet cells. So they have goblet cells that produce mucus, and this mucus gets secreted onto the surface of these ciliated epithelium. And the ciliated epithelium they either in the respiratory tract move the mucus along so that when you breathe in a bacterium, it's going to move anything trapped in that mucus. You're going to swallow and it's going to be thrown in the acid pit of the stomach. So that's just going to be, just like in olden days when they threw things into acid, yeah, we still do that. So we throw things into acid, we trap them in mucus, we pull them along, we swallow it, throws it into our stomach, and they just get dissolved, okay? So this is how we utilize this mucosal epithelium. And the mucosal epithelium is, again, if you think of the fort that we're in, you have to think of the fort that we're in as this big, thick wall, this wall with bricks and cement that's this big and this wide, and on the surface, here we have, because it's not in your books, these are stem cells. We call them cuboidal stem cells. And in your skin, you always make more skin. Have you guys, when you guys go to Marseille and you get burnt, what's going to happen to your skin? It's going to peel off and you're not going to have any more skin anymore, right? No, of course not. So these cuboidal epithelium, these small square, are your stem cells. And they grow and divide, and when they grow and divide, they always push up. And as they push up, they start the process of activating the production of keratin inside the cells. And as they become keratinized, they become flattened, and keratin is a waterproofing molecule. So not only that, on the top, as they move up towards the top, they're dead. So all over the surface of our body, we are covered in a layer of dead tissue. And this is important, specifically against something that has to grow in a living cell, such as viruses. Viruses have to grow in a living cell. So if we cover the surface of our cell, the important bits with a dead wall, they have nothing to grow and reproduce it. So what's again the word? We block them. What's the real word that you guys use? I'm just trying to remember. What do you do when you spike somebody's, it'll come to me. Anyways, yeah, I know. It's whenever, you see again, it's not that I don't know your guys' names, it's just that when I see you on the street and I'm not expecting to see you, my mind immediately goes blank. So that's just what's gonna happen, I'm gonna warn you. So this is, again, we have keratin, and then we have secreted in the gastrointestinal tract, and this comes from the book, okay? So I'm just putting images to what the book is saying, okay? So here we also have tight junctions. And tight junctions are when your skin peels, is why your skin peels as a sheet, right? It's because all of those tight junctions, and this is why leather is so impervious to everything, because that's the skin of a cow, right? Because you can't tear leather because of the connective tissue underneath. So you have this layer of connective tissue, you have the surface, you have all of these other things that make this a tough, impervious thing that organisms can't get through. Unless there's a breach, right? What happens if there's a breach? Cut. There's a breach, things have gotten through, right? So, how long does it take for you to know that you've gotten a paper cut? Instantaneously? So, we are going to have all sorts of sentinels in our IELs, interepithelial leukocytes. We're going to have all sorts of there's going to be specific forms of dendritic cells that are in our skin that are sentinels for as soon as the wall is breached. As soon as the wall is breached, you need to make sure that that wall, that you pour in troops to stop the influx of invaders, right? So this is what we're going to do. And again, epithelium also produces these antimicrobial substances, e.g. defensins and cathelicidins, okay? That kill microbes in infected cells. So, if that's all I want to talk about, just remember that the reason that this is thin is because you have to be able to take in food and nutrition and water. And this allows you in the gut to take in food and nutrition and water. in the lungs, it allows you to take in oxygen, right? It allows things into your respiratory epithelium, capillaries right underneath here can take in oxygen. So we can't have it a big, thick layer. And the other thing is when we talk about this epithelium, the stratified squamous, we have stratified squamous epithelium all along the outside of the body. And I tell people, this is Kim Kardashian, okay? There's a big butt here, and then a tiny little brain over here, okay? So, even though she's becoming a lawyer, what? Okay. Really? Who's taking the test for her? Okay, so, I hate the Kardashians. I hate all of them, okay? Anyways, the thing that's important is on the areas of the skin is most at risk, most likely. Remember she talked about building the fort up on the Bastille, up on the mountain, because most likely the invasion was going to come from the mountains, because that was the closest area and was going to come from that way. So we made our defenses stronger in that area. Just the same thing is true on calluses, on your hands and your feet, are really, really thick walls of stratified squamous epithelium, because our defenses are always we're always having things try to get in, always trying to be breached. So we have thicker skin there. The same thing is true if you have a brother who ever pinched your skin underneath your arm. Very thin, we call chin puppies. They hurt and you bruise like crazy because your skin is so thin. I could pinch this like this and have nothing happen. Pinch this underneath your skin, the skin is very fine, because it's mostly always protected, right? Because this is one of the things. So we make the skin thinner where you have other things that are protecting you, we make it thicker where we need it. With the mucosal epithelium, all we have, or it's columnar, and a lot of times you will see it called pseudostratified columnar epithelium, because when you actually see it, in order to get one layer of cells, these nuclei are big, and so you have to put a nuclei here, a nuclei here, a nuclei here in order to fit them into one layer. So it looks like there's 3 layers of skin just because the nuclei are at different levels, right? But there's still just one. Does that make sense? Goblet cells are what make the mucus. Goblet, they produce the mucus. We're going to ciliate epithelial cells work together, and for people who have cystic fibrosis, their ciliated epithelial cells don't work together in something called a mucociliary escalator. They kind of just go like this, and so they don't move the mucus around. So we're going to talk about the toxic molecules we put in our mucus, but if the mucus isn't being pulled along, when they breathe in pathogens, and there's pathogens in the grass out there, there's something called Pseudomonas aeruginosa that's just in the dirt. And for you and I, Pseudomonas aeruginosa is not a problem, because our ciliated epithelial cells move it along, we swallow it, and it gets destroyed. For people who have burns or problems where their skin their defense has been peeled away, or people who have ciliated epithelium that doesn't move, so it just kind of jiggles a little bit, the mucus stays there, the bacterium get in, and they can grow in everything. Pseudomonas aeruginosa can actually grow in disinfectants. It's that good. It's a horrific pathogen if you're immunocompromised. If your skin is burned, it's one of the things that will kill a burn victim, because it can get on the skin, it can colonize, it glows green, and it smells. So when you see a burn wart, it's in the olden days, it used to have that terrible smell. It actually almost glowed green because the Pseudomonas aeruginosa would just get in, colonize, and it's a gram negative. So it also created endotoxin, so it just adds fire to the whole process. So these are the things that we worry about. These are the things you and I are protected. You and I can walk out, roll in the grass, and have no problem. I have a student who has cystic fibrosis, and she worries. She knows about every organism that's in her lungs. You can tell what's there. They do scopes of what's in her lungs. And one of the things she's most worried about is Pseudomonas aeruginosa. And so she has a defect in one of her cilia, so that they just don't move the same way. Or there's all sorts of different genetic defects. But genetic defects on any level of your immune system on any level of your skin. If you have a burn, if you're stupid enough, in North Carolina, we're kind of dumb. If we beat a team called Duke University, they all go out onto the street called Franklin Street and they jump over burning couches. And I actually know the head of the burn center works in my department. And he's gone on every time there's a Duke game, he goes on and said, do not jump over burning couches, because this is a long lasting, a burn can kill you. Because once it gets infected and it's going to cause pain and it's going to suffer, if you just don't jump over the couch, don't go near the fire, okay? Don't die in a fire. In World of Warcraft, that's one of the most important things. Do not die in a fire, okay? So you do not want to stand in the fire because it's going to take your primary defense away. Okay? Any questions about this? Do you have any questions about any of this? Okay. Now we're going to talk about the other cells of the innate immune response. And we're going to go a little bit more in depth, just a tad, because I don't think they give PMNs or mononucleophagocytes the love that they deserve. These are my boys. Okay? That's my jam. So, all I can say is that without PMNs, you guys would not be here right now. Without functioning PMNs, you would have died in childhood from overwhelming bacterial infections, extracellular bacterial infections. So write a big, only extracellular bacteria. That's all they're good at. Only extracellular bacteria. These are the two main phagocytes of the immune response. Okay? So, the other name for PMN, and PMN stands for, and this is in your book, so I'm not going to write it out, polymorphonuclear cell. So, let's look at this cell, and let's draw a line around its nucleus, shall we? That's what the nucleus looks like in this one particular cell. Guess what it looks like in another cell? Different. Thank you, Haley. It's different in another cell. They are polymorphic. And the reason that they're polymorphic is we want a long, sinuous nucleus, because just like we were talking in the ciliated epithelium, those nuclei are big and fat. So if you need to slink through something, if you need to be able to slink your way through something, you need to be able to mush yourself through. Have you ever seen a mouse get underneath a door? They can literally bend their whole muscular skeletal system and get through something that's literally like this big. And so we need a cell that can do that. Whereas, what do you think about this big old fat nucleus there? How easy is that going to get in? It's like me trying to get in a size, anything, we'll say size 2, okay? It's not going to happen, okay? So, we have these two different kinds of cells. We call them, and again, PMNs are part of the family, part of the cells that are called granulocytes. And the reason that they're called granulocytes, Anna, can you guess? You can see granules in them, okay? Because they're granulocytes, and the 3 granulocytes, they don't talk about the other granulocytes that we're going to need to talk about in the future, so I'm going to write them out here. So there are 3 granulocytes that are all come from the same precursor, right? So that, and I did notice that finally they actually talked about a hematopoietic stem cell in chapter 2. So they talked about the hematopoietic stem cell in chapter 2. When we talk about PMNs, PMNs are also, see there's a polymorpho nuclear cells. We abbreviate that PMN. But another name for them are neutrophils. Another one, so this is the primary one. This is the most important, star, star, star. So, remember, when we were looking at immune response, and it wasn't until it wasn't until one of our Nobel Prize winners learned, they were the ones who discovered how to do gel electrophoresis, and he won a Nobel Prize for that at UNC. So it wasn't until he started learning how to do that we identified these globulins, these proteins, these globulins that we found in immune response, and we called them immunoglobulins. So when we were trying to stain, when Christian Graham started Gram staining, we could stain one bacteria pink and one bacteria purple. Somebody else started thinking, well, can we do that with the cells of the white blood cells? Can we stain them differentially? So they started looking at how to identify the different cells of the white blood cells. And so they did see that there were granules. When we stained certain cells, it appeared that there were granules in their cytoplasm. One set of cells stained with a neutral dye. So we call them a neutrophil. One stained with a basic dye, and so we call them basophils. One stained with a acidic dye called eosin, and so we call those. So, if they stain differentially, do the granules contain the same stuff? Of course not. So that means every cell has a different set of granules for a different reason, right? PMNs are the only ones that are phagocytic. PMNs are the only ones that can eat things. These cells degranulate their granules in order to make their effector, the function, okay? Does that make sense? Okay, so we have eosinophils, basophils, helminths. These are primarily against parasites. Mm-hmm. William?
Neutrophils are the only one that can phagocyte.
I mean, the only one of this set.
Yeah.
Yes, because in a lot of places it's inappropriately stated that eosinophils are phagocytes. They are not. Okay? They just degranulate. But their response is to parasitic worms. And in the United States, we hardly ever have parasites in the United States. In Africa and in India and in any third world country, without these cells you would die. Okay? So again, it depends on where you are globally, what kind of access you have to clean water and flush toilets. Flush toilets have made the difference in the world of health and safety. So this is one of the things. So polymorphonuclear cells are neutrophils, interchangeable. I will see a paper where they discuss PMNs and neutrophils off and on. Okay? Does that make sense? So it's like calling an automobile a car. Okay?
Can neutrophils also degranulate, or do they probably?
We're going to talk about that. They are so cool. They actually don't, they do degranulate, but we're going to talk about that just in a little bit, okay? As a matter of fact, almost right now. So the other thing is that when we're looking at a white blood cell count, so if you watch Grey's Anatomy and they ever talk about a CBC, okay, complete blood cell count, okay, they're going to look at what is normal. Normal is you have different amounts of these cells, and PMNs, the book gives you numbers per mil, which makes no sense to you. Because who knows what numbers per mil you have, okay? So in phagocytes or PMNs they're about 60 to 65% of all your white blood cells. When you do a white blood cell count, and if any of you have ever done a drawn blood, I used to draw blood and do HLA typing for a living. You can draw blood, and when you draw the blood, this is red blood cells. Red blood cells are the heaviest, and they sink to the bottom first. And then on the top of this is something called a buffy coat. And a buffy coat is where all the white blood cells are. And what do you think this is? Plasma. Or if it's in the body, what would it be called? Interstitial fluid. Okay? Okay, so
we have this buffy coat, and if we took, and I would suck the buffy coat off and throw out 65% of them, because all I was looking for was lymphocytes or antigen presenting cells. So PMNs were no good to me. So I got rid of 65% of them. However, if you're doing a complete blood cell count and all of a sudden you see 85% PMNs, what does that tell you? You have a bacterial infection. What happens if all of a sudden, normally eosinophils are about 3 to 5%. Whoops, no, I'm writing on with my, about 3 to 5%. What happens if there's 15%? You have a parasitic infection. More, and we're going to talk about this when we get to allergies, these are involved in allergies and asthma. Okay, so we're going to talk about this more. Basophils, 1%. Okay, so people hardly ever discuss basophils. Okay, mononuclear cells, about 10 to 12%. Okay, and again, you can make that so it equals 100, depending on if you have more PMNs, you're going to have fewer probably lymphocytes, because if you're having an extracellular bacterial infection, what cell do you not need at all? T cytotoxic cells, because can T cytotoxic cells kill a bacteria? No. So the immune system shuts down the production of those, ramps up, and releases. In the book, it talks about how many PMNs you have in your body. When we release certain cytokines that we're going to talk about in just a little bit, a specific cytokine causes the bone marrow to release a flood of PMNs into the system. Just releases them into the system immediately. Millions and millions and millions of them, because bacteria can grow exponentially. So we're going to flood the troops. We're going to open up the doors. We're going to put as many troops out there to eat as much as they can. Okay? So we're going to flood the troops out there. The other thing that PMNs are going to do, so they're going to eat their phagocytes. They eat primarily extracellular bacteria. We're going to talk about what's in their granules in just a minute. We're also going to talk about, what was the other thing? About how short-lived they are. They live 1 to 3 days. So we are constantly, because if you think about this, when we're making PMNs, we are constantly, they are the most numerous white blood cell. So we have to make 65% of all our white blood cells every day. So we're constantly making more PMNs. So our stem cells are constantly differentiating into PMNs. And then we make reserves for the bone marrow, and if those don't get used, they die. That's how important it is that we have enough PMNs to fight off a bacterial infection as soon as it happens, because they can outcompete us. So we're going to have PMNs. They're going to live 1 to 3 days. If they're not used, they're dead. If they've eaten and thrown away all their granules, then what they do is something called a neutrophil net. And what they do is I've done everything I can for you. I'm going to literally self-explode, and it explodes, and it throws its DNA. And remember, every cell in your body has all 3 billion base pairs of genetic material in every cell. Your butt cells have them, so if I wanted to clone Brad Pitt, all I would need to do is take a little, you guys don't know who Brad Pitt is anymore, but okay, so think of who would be good. Chris Hemsworth. That didn't work. Let's do that, okay? So if I wanted to clone Chris, just take a little stab, take those nuclei out, and I could use his genetic material to make a new one. Unfortunately, it would go through the embryonic stage, and then he'd be a baby, and he'd be useless to me, and then I'd take care of him, and it would be like mine. Not what I'm looking for. Okay, so I'm going to erase that off of the tape. Okay, so but these, they're going to make a neutrophil net, and then what they do is they self-immolate themselves. They throw their genetic material out into the environment, and DNA is sticky. Because DNA is sticky, bacteria get trapped in it. So even if they can't eat anymore, they throw their genetic material out, causes a net, and then there are receptors on the surface of this genetic material. Because they're trapped, we can then bind to them and eat them up. Other things can eat them up, like macrophages. Okay, does that make sense?
So the receptors are binding
You don't need to know.
macrophages?
No, so you don't need to know what the receptors are and how they recognize, how they get, probably the bacteria in there. We're also going to talk about this when we talk about lupus. Okay? We'll talk about this in more detail about this in a little bit.
So you're saying that the genetic material catches the bacteria and other things come and destroy it after the PMN.
Because the PMN is dead.
Yeah.
It has destroyed itself, right? So maybe other PMNs are around. I don't exactly know. We just talk about neutrophil nets. I haven't actually read any papers on exactly the mechanisms, neutrophil nets. So I'm hand waving, jazz hands here. La la. Don't know. Okay. Probably macrophages, because macrophages are supposed to have the garbage men of the immune response. So the other thing that's important about these mononuclear phagocytes, that these are also called, so the precursor cells are called monocytes. And these monocytes can differentiate depending on the need into either macrophages or, believe it or not, dendritic cells. Okay, that's a really cool slide. Okay. Both macrophages and dendritic cells are phagocytes. Those are the only three phagocytes of the immune response. Okay? Are we good? So if I ask you to name three phagocytes of the immune response, what would you say? Would you say eosinophil? No. Would you say basophil? No. Could you say neutrophil? Yes. Could you say mononuclear phagocyte? Is that? No. Technically, that's two, right? So you need to explain both macrophages and dendritic cells. And the other thing the book talks about, let's see what the next slide is. Oh, I think I've already seen the next slide. So the other thing the book talks about is what happens in these mononuclear, monocyte dendritic cell precursors. Monocytes are the cell that we just talked about. Monocytes can become macrophages or dendritic cells. They can become either, okay? So those mononuclear phagocytes are now lumped into, again, it used to be that we thought if you were macrophage, you always became a macrophage, you always were a macrophage and that's all you could be, and if you were a dendritic cell, that's all you could be. Now we know that it's kind of fluid. If you need to move back in towards a dendritic cell-like state, you can. Okay, let's take a break and come back and we'll finish this up. We'll come up, so everybody take a break, walk around, come back in three minutes. Go to the bathroom, 5 minutes if you don't get back in time. Okay, you guys, we're going to get back to this. Oh yeah, you guys, we were talking about, remember to bring stuff for picnic, food, for whatever you want to eat or share for Friday. We're starting to talk about this. And I was on the tram on the way through, and do you remember that big glass building we went through? She said it was a covered market. You should have seen what it looked like this morning. If you want to get up a little early, like I got up at 8 to go out and kind of shop around, it would be fun. You guys, it's an easy 10-minute tram ride from here to that. And you don't have to go all the way to San Bruno in order to get a decent market, where she was talking about how, and it's beautiful inside. You can just take pictures of everything. So anyways, I just wanted to remind you guys about that. So, because Chris and I were already talking about this.
Oh, she doesn't. Because I have a question.
Oh, no. Let's talk about this. Yes. Okay. We're good. Let's all go back to immunology now. Okay, go ahead.
I just wanted to know if monocytes are still considered differentiated cells if they do transform in macrophage.
So that's a great question. Monocytes, I don't consider a monocyte a differentiated cell. A monocyte is always a precursor cell. You never see really the monocyte doing anything. It's going to differentiate either into a macrophage or a dendritic cell. And it can differentiate, let's write dendritic cell in there, because it can actually, not just macrophages, it can differentiate into anything or DCs. We'll put, the way that we abbreviate dendritic cell is we call them a DC. Okay? So they can, depending on the milieu in the area in which they find themselves, the inflammatory response, they will differentiate into what the immune system needs to be. Please?
So if the dendritic cells and macrophages come from the monocytes, how does the mononuclear phagocytes relate to those?
Technically, they're all the same terms for all the same cells. So mononuclear phagocytes, it's just another way of saying. So mononuclear phagocytes are monocytes that become dendritic cells or macrophages.
So monocyte is the same thing as mononuclear phagocyte, basically.
Monocyte is the precursor cell to those. Okay, does that make sense?
Okay, so that means the dendritic cell and macrophages are both nuclear, mononuclear phagocytes. Okay.
Yes.
I think it's just differentiating between polymorphic nuclear cells, like it's like the two kind of, one has one has one.
Oh yeah, exactly. That's exactly. Yeah, because yes. And the thing is again, it depends on, and again, monocytes are not granulocytes, even though they're going to have lysozymes in they don't stain, and they're from a different pathway of differentiation. And so all of them come from the hematopoietic stem cell, and then from the hematopoietic stem cell, everything diverges into different pathways, okay? Granulocytes diverge into PMNs, basophils, and eosinophils. Also, we're going to talk about another one that we did not talk about there because we're talking about circulating. We'll get to another granulocyte that's really, really important, and we haven't talked about it yet because it's weird and it's different from any of those. So we'll get to it, okay? I know. Whoa. Okay, so, yeah.
Sorry. So monocyte is the precursor to dendritic cells and macrophages, and dendritic cells and macrophages are both types of mononuclear phagocytes.
Correct. Perfectly well put. Okay? So really, again, so a Beamer and a Jag are both forms of cars. Okay? Different forms of cars. Lovely forms of cars. But they're both cars, right? They're just differentiated forms. Okay, does that make sense? And then all of them are cars. Passenger cars. Whereas we have a drag racer car that's different. Okay, does that make sense?
So, are the eosinophils and basophils considered as PMNs? Or are they?
No, they are a form of granulocyte. So when we draw, as a matter of fact, if you go back to, I went ahead, shoot. Where did I put it? I put it on the other PowerPoint. I'll show you. On the first PowerPoint, I linked, when I put hematopoietic stem cell under that first PowerPoint, I linked it to Wikipedia because Wikipedia is actually fabulous. I love Wikipedia. It's where I go to if I don't understand something. Because most of the time, I always talk about my people. My people. If you've seen The Big Bang Theory, those are my people. Okay, scientists, we're all on the spectrum. We are obsessive about our topic, and we will go in and make sure that anything that's on Wikipedia is up to date, and you want to be the one who puts the information in and says that that's your sentence that's still there. My nephew went in and said he was dating an actress for 20 minutes until it was taken off her profile page. So you can go in and change Wikipedia, but most of the time bad stuff is taken out almost immediately, okay, or incorrect stuff. Okay, so, yeah. He's dating Mila Kunis for a while. Mm-hmm. Yeah, I know. At least in his own mind he was. Yes, exactly. So, he has never met her, just so you know. Okay, so, the other thing that the book talks about is there are all other kinds of macrophages throughout the body. And all of these other macrophages throughout the body have a different, they're not mononuclear phagocytes. They don't come from monocytes. They come from this precursor hematopoietic stem cell. Here we have a hematopoietic stem cell. And they migrate during embryogenesis. They migrate to the tissues where they differentiate into macrophages that are vitally important for keeping your health and your well-being. So we have microglial macrophages in the brain. We have Kupffer cells in the liver. We have alveolar macrophages in the lungs. And we have sinusoidal macrophages in the spleen. So those are different forms of basic macrophages. We're going to talk about a different form of this kind of differentiated dendritic cell. So we have a dendritic cell that can come from a monocyte. And we call those mononuclear phagocytes, or those are cells that have differentiated from a monocyte. We're going to talk about something called a Langerhans cell. It is just a dendritic cell. It's a specialized dendritic cell that doesn't come from them. So that's why we differentiate these. So we're going to talk about these in just a minute. So here you have the monocytes and the macrophages, and these tissue, and they are vitally important. Your liver filters everything, and so we have to be able to clear the stuff that we're filtering out of the liver, and the macrophages do the job. You can think of them as the garbage men of the cell. If a cell dies, they're there to eat it and clean it up and destroy it. So they have a lot of toxic molecules that we're going to talk about in just a minute, okay? So we have macrophages. And again, when we talk about these phagocytes, all phagocytes need the ability of destroying things they've eaten. And if they can't destroy the things they've eaten, we have disease. So just like we saw, if your ciliate epithelium can't work, you're going to have disease. If your skin is breached, you're going to have disease. Everything, every aspect of the immune response, if one small aspect of it doesn't function correctly, you will have more disease. We're going to talk about this in just a minute. But here, we talk about these are the receptors that are on the surface of phagocytes. We're going to talk about complement in just a minute. Probably I'm going to assign you complement to go over for tomorrow, but we'll see what we get to. All of these molecules have CD numbers, and depending on what CD profile they have, which complement receptor they're expressing, we actually differentiate macrophages and dendritic cells from monocytes into the different CD16 that they express. So again, the different complement receptors they express help us identify if it's a macrophage at this point in time or is a dendritic cell. So we have names for these, and so these receptors are vitally important because when they bind these fragments of complement breakdown products in bacteria, it activates a pathway of signaling into the organism that says, I need to eat this and I need to destroy it. And the way that we do this are we have this activation. So this is going to not just drive NF-kappa B. So I want you to know that that transcription factor profile that gets activated goes like this, poof, okay? And so all of these things are being activated from those toll-like receptors. We're going to have NF-kappa B that's going to make, it's going to activate the production of pro-IL-B1 or IL-1 beta, right? So that's one of the things that it does. It's also going to activate the upregulation. So we already talked about IL-1. I want you to know that these are the three main pro-inflammatory cytokines. So you guys will need to know. This is one of the 12 that you need to know. So TNF, IL-1, and IL-6. Okay? These are the cytokines that make inflammation. And I have never read a paper where one is discussed, where at least one other one isn't discussed in the same paper, because they come generally as a set of three. If you're going to have IL-1, you're going to have TNF. And again, as a pharmacist, if you're going to look at analgesics, something to stop fevers, or something to, what Tylenol does, you need to know these pathways, you need to know what these things are. And as a matter of fact, for some things, we're actually making medical therapies to help stop inflammatory responses. For people who have excessive immune responses, we actually make antibodies that bind to and soak up some of these molecules to reduce inflammation, because it's for people who have any sort of autoimmune diseases, their inflammation might be out of control. Inflammatory bowel disease, their inflammation is out of control. So these things we need to figure out how and what to do. And what people say, well, why can't we just make an antibody to LPS? We've tried and it doesn't work. So everything that you can think of, probably we've tried and it doesn't work. So, but we can bind to these things, soak them up, and keep them from causing such a huge problem or huge role. We're also going to talk about IL-12. IL-12 has a specific action on activating specific cells of the immune response. In just a minute, we're going to talk about that, okay? But then the other thing is, so we're going to make these, so one of the things that is made is generally, these things are going to activate inflammation, and they're going to respond to, they're going to make enhanced adaptive immune responses. Two enzymes that you need to know in a phagocytic response are vitally important. We're going to make something called nitric oxide, and so has anyone heard of, so whenever you eat yogurt to stop what? I mean, what are the bad things that happen that you eat yogurt or good things to offset? Has anyone heard of like oxidative mediators, or what's the word I'm looking for? Again, I need to think of it. I know what it is. I've used it 1,000 times, and my brain just went, boop. What is it when you have free radicals? Have you heard of the term free radicals? Or, so free radicals, any time that you are using, bacteria can make them, we're actually creating free radicals with this phagocyte oxidase. And phagocyte oxidase actually creates these free radicals. It's taking an oxygen molecule and it's making hyperreactive. It wants to bind to something. It wants to bind to DNA. It wants to bind to proteins. It wants to bind to lipid. And anytime it does, it destroys them. So when you eat yogurt to help stop free radicals, or something like this, so they're saying that those things can help reduce these things. We are creating them in our white blood cells, because when we eat something, we have to throw those toxic reaction molecules at them so that they can act on the bacteria or the virus or whatever it is in there to kill it, right? And so we're going to have phagocyte oxidase, so that creates reactive oxygen species, also known as ROS. Okay. There's RUS, and those are rodents of unusual size, if you know The Princess Bride. Okay. This is ROS. Okay. So we have reactive oxygen species, and then we have INOS, and INOS is also a molecule that's actually used as a signaling mechanism in all sorts of things. If you guys have had physiology, you know how nitric oxide can do all sorts of amazing things, but it can also is also a toxic molecule that acts to kill microbes. And so we're going to have this is going to bring in the thing that we're trying to destroy. We're going to throw toxic molecules at it, and these are the two main enzymes that create the two main toxic molecules that allow the killing of this. Okay? So then, we're going to talk dendritic cells. Any questions about macrophages or that pathway is primarily found in PMNs, macrophages. Dendritic cells are generally their job is primarily to be an antigen presenting cell, to activate the acquired immune response. Now we're going to learn that other cells can activate the acquired immune response, but dendritic cells, that is the only thing they do.
You said it was a phagocytic cell? What makes it a phagocytic cell?
It can eat things. But we'll talk about how and why in just a minute, okay? So that's a great question. So they are different than macrophages. Macrophages always eat things, Karen.
So when you say PMNs, like that pathway within PMNs, is it just neutrophils or all PMNs?
So all PMNs are neutrophils.
Okay.
Those terms are interchangeable. Eosinophils and basophils aren't phagocytes.
Okay.
So they can't eat things. So it's PMNs, macrophages that primarily have that toxic molecule to make those toxic reactive oxygen species, okay? In the granules of eosinophils and basophils, and the other one we're going to talk about in just a minute is mast cells. Those granules are different, they have different functions. They are full of different active ingredients. Okay, does that make sense? When we talk about dendritic cells that are part of an embryogenesis, they are not from the mononuclear cell line. These are so vitally important, they're called Langerhans cells. And Langerhans cells are the ones that are interdigitated into the stratified squamous epithelium. So when we talk about the wall, in order to have a wall that's functional, if you don't put any sentries on the wall, then all you have to do is scale the wall, get over the wall, and you're in, right? So you need something that says the wall has been breached, the wall has been breached, the wall has been breached. And so you can kind of think of this as, I like to think of this as, have you ever seen those shows where you have to get through a laser? You know, there's laser, 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 and they have to climb over this, and then crawl under this, and then, but most of the time there's so many things that they're gonna trigger something. The same thing is true with the dendritic cells. If you think of a dendritic cell as something that looks like this, like a dendrite, okay? And it's interdigitating into all of the cells in the stratified squamous epithelium. It's interdigitating into them. It's not bound by tight junctions. They're just wedged in there, okay? And in this state, they're immature. They are an immature dendritic cell. All dendritic cells, until they're triggered to activate, are immature, okay? All dendritic cells. Macrophages that haven't been activated by pathogens are just macrophages, they're just garbage men, okay? So what happens is if you can see these, imagine something cutting here and bacteria trying to get in through any of these laser beams that are identifying what's going on. And then so we have both dendritic cells in the epidermis, and then we have dermal dendritic cells as well. But then we also have dendritic cells, so when things get pulled into the lymph nodes, we'll have dendritic cells waiting there as well. So we have dendritic cells that are waiting in the lymph nodes so that if it gets in through some place that maybe there aren't as many Langerhans cells, it gets into the respiratory tract, and and the alveolar macrophages let this get through, then there's dendritic cells in a lymph node to activate the T cells immediately. If something gets pulled in by lymphatic fluid, like a bacterium or bacterial products, we can take that, the dendritic cells are right there to start activating the acquired immune response. They don't have to migrate. These are the ones that take about 24 hours to migrate to a nearest lymph node because they're immature. And they have to mature. And the interesting thing, Elise, is this answers your question. What they have to do is they are only phagocytic in their immature state. So they phagocytose something that triggers their PRRs. Can anybody name a PRR? A pathogen, pathogen recognition receptor. A TLR. Very good. A PRR is a TLR, an NOD, and RLR. A pathogen, those are the names for those things. So on the surface of dendritic cells, on the surface of Langerhans cells, all those toll-like receptors are there, and all of the dendritic cells are just hanging out in the skin. I'm just hanging out, these Langerhans cells, I'm just hanging out. Something gets in through the skin, triggers their PRRs, their toll-like receptors, or their NLRs, or their RLRs, or their complement receptors. It's going to initiate them into an activation process where they pull themselves out of the skin, they round up, they drop into the nearest lymphatic vessel, and they move like hell to the nearest lymph node, where they're going to activate a immune response, an immune response. Okay, does that make sense? So, they're immature. And we don't want them to be phagocytic once they mature, because on their way, they're going to be passing by what? Self cells. We don't want them eating self cells along the way, along with the pathogenic, because that could accidentally trigger an autoimmune response. So they're only phagocytic initially, and then they have this migration pattern. As they migrate, they mature into professional antigen presenting cells. So in the 24 hours it takes them to get from the skin to the paracortex, they have gone from an immature cell into a mature cell, a mature antigen, professional antigen presenting cell. And if anybody's seen Lord of the Rings, again, I am a geek, you know that Gandalf goes from Gandalf the Grey, he's a low level wizard, to Gandalf the White, after he goes through the activation process where he fights off this big demon, okay? Where he's got all of a sudden these superpowers. That's a dendritic cell that's been activated. They have superpowers. They are officially told you can activate the acquired immune response. You can have the key to activate weapons of mass destruction. And they do. Not until they're activated by some sort of pathogen, right? So self isn't going to activate these. Only something that's going to trigger something that binds to LPS, bacteria, something that has double-stranded RNA, viruses, something that has flagellin, bacteria, something that has bacterial lipoproteins. Mm-hmm.
So are dendritic cells still phagocytic when they're in the lymph nodes already, or are they?
These are immature in here
So
until they get activated by PRRs. Until something drains in there that binds to their PRRs, that activates their activation process. They still have to go through the activation. They have to become a professional antigen presenting cell.
So the only time it's like you have to worry about them picking up peptides is when it's moving through the body?
So we're going to explain this when we talk about cancer. When we first, in quick overview, when we first started trying to figure out how the immune system works, we're going to talk about something called MHC. MHC is difficult because every single one of you have different things that you're going to put in your MHC molecules. So your cancer might be displaying different peptides than your cancer. So what we thought is, well, let's just take these immature dendritic cells, these mononuclear cells, make them into dendritic cells, feed them cancer, activate them, inject them into you. They're going to migrate into the immune response and activate the correct T cells. And so we did that. So we took the cells out of people, their own cells, with their MHC molecules. We activated them into fully professional antigen-presenting cells while we fed them cancer from the tumor. And then we injected them back into the person. And do you know what they did? Nothing. Because they had already gone through their migration pattern. They were already professional antigen-presenting cells. And the migration pattern only happens when you're an immature moving towards a lymph node. You're looking for those chemokines that's drawing you to CCR7 because it upregulates CCR7 because that's where the T cells are. So once these dendritic cells were fully functional, they then downregulate that because they should already be there. So they didn't migrate anywhere. So we had to figure out how and when to give cancer proteins to immature dendritic cells and then put them back into you as quickly as possible so that they had the migration pattern so that they appeared to be doing what they normally would do. Did that make sense? Isn't that cool though? So we learned. So unfortunately the people with breast cancer who had the initial studies did not.
So the way that they present antigens is by phagocytizing
whatever.
Oh, that's coming. At least that's coming.
Okay.
You're just going to learn that they're going to be able to present antigen. Okay, it's coming. Chapter 3 and chapter 4. If you want to read ahead, chapter 3 and chapter 4. All about that. Okay? So these are dendritic cells. And then the last thing is again, so dendritic cells are antigen-presenting cells. Langerhans cells are one of the best antigen-presenting cells we have. They are not monocytic precursors. They're embryogen. They come from a hematopoietic stem cell. They migrate to the skin and they differentiate into Langerhans cells there. Okay? And they just divide and they keep. We don't even know how they keep dividing and how they keep. We don't always understand because people aren't investigating them. It's very hard to study. Mast cells are also only starting to be studied. Mast cells are the granulocytes, the innate granulocyte that's only found in tissues. But it's found in tissues wherever there's vasculature. So where is there vasculature in your body? Any place there's living tissue, right? So every place there's living tissue, there's going to be mast cells around the capillaries, around the arteries, around these things, because they're sentinel cells. If something gets through and triggers a mast cell, they degranulate to cause an inflammatory response. Okay? So, the other thing that we're going to talk about, and I'm only going to briefly talk about these because we're going to come back to these, but I wanted to introduce these to you. These will not be on the exam. These cytokines will not be on this exam. These cytokines will be on the second exam. Because the weird thing is these innate lymphocytic cells are exactly like acquired lymphocytes, T helper cells. This is just like a Th1, this is just like a Th2, this is just like a Th3. Same transcription factors, same transcription factor, same transcription factor, same cytokines that induce, cytokines that are produced, same responses. We never knew they existed until about 5 years ago, maybe 10 years ago, because time goes by so fast. So we finally found out where the source of the IL-4 was. They were from cells that this doesn't look like, it doesn't have a T cell receptor on it, doesn't look like a T helper cell, but my gosh, it's making IL-4. What is it? So they started investigating these things, and then they are exactly similar to, they are the analogs of the acquired immune cells. So we're going to come back over these. We're going to take this, and we're going to put this under the T helper cells, and they're going to be exactly the same, only they're called an ILC1 and ILC2 and ILC3. They're exactly the same. The same thing is true with NK cells. We're going to talk about NK cells. NK cells are the T cytotoxic cell innate analog. They kill human cells. That's their job, is to kill human cells. They throw something called perforin and granzymes at it, just like T cytotoxic cells do. So all of these things we're going to talk about. Immunity to viruses, intracellular pathogens. Immunity to cancer, a self-antigen that is run amok, a self-cell that is running amok. You don't have cancer from other places. Cancer is you not functioning correctly. Okay, does that make sense? Okay, so these, I just wanted to show you this now because we're talking about the innate immune response. William?
So does that also trigger specific responses, or is it like more?
So we are going to talk about that when we get to those things, okay? And those are the rest of the cytokines you need to know, okay? We're going to talk about IL-12. We've already talked about IL-12. We're going to talk about IL-22. You guys aren't going to need to know IL-33. You're not going to need to know TS, only things that have IL in them, okay? You're not going to need to know those other things, okay? But this is the innate lymphocytic cells, ILCs, that are the analogs of the acquired immune response. And we think they clonally divide, just like the acquired immune response does. So if they can clonally divide, and if they're responsive to specific triggers, how is that innate? I mean, so they clonally divide. They say that these cells don't clonally divide. Did you guys look up NK cells? Do NK cells clonally divide? So I'm just going to give you a brief heads up. NK cells can clonally divide, and we think their innate receptor gets modified to be more specific the more experience it has. We think the same thing is true with mast cells, but again, that's above your pay scale. So what we're learning is everything that we thought we knew about the innate system, people didn't study, and once we started studying, we found out that we were wrong, or not as clear as we needed to be. So here, we're talking about an NK cell. So an NK cell, and they introduced this in this chapter, so we need to talk about it before we even talk about MHC molecules. MHC molecules are the display molecules for T cells. So, without a display molecule, a T cell can't kill, okay? But it's specific for the peptide that's being displayed. For NK cells, it's different. NK cells want to kill everything. They want to shut shit down. They want to kill something. That is what they're programmed to do. Okay, so what they come up to, and every cell on the body, and they're triggered, they look for every cell in the body that has a nucleus in it. Every cell except red blood cells express these MHC molecules, this one flavor of MHC molecules. Okay? When a virus gets into a cell, a virus can shut down that MHC molecule, because if there's no MHC molecule presenting viral peptide, T cells can't kill them. You can't see us. So what they do is, so if there is an MHC molecule, if there's a normal cell, we're going to leave that to the T cytotoxic cells. So NK cells have two signals. They have, every cell in the body has an activating signal for NK cells. So the first signal for NK cells is to activate it to kill. You get to kill, 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 and then if it binds to an MHC molecule, I can't kill. Okay, I'm going to leave this cell alone. MHC is there. I'm going to bind to this, and I'm going to kill, kill, kill. Oh, MHC is there. I can't kill. Okay? I'm going to bind to this, kill, kill. No MHC. That means something is wrong with the cell. Something is screwed up, forked up. Okay? If it's forked up, it needs to be killed. Okay? So we will kill it because there's no MHC molecule to tell me I can't kill it. And that's what NK cells do. Does that make sense? So, if the MHC molecule is lost, that allows killing. If the MHC molecule, so it's a two-step signal. Step 1 activates, step 2 inhibits. If step 1 just happens, the NK cell is activated to kill, which is exact opposite of how we're going to activate T cells. It's the exact opposite. It is the yin to the yang of T cells, okay? Anna.
Okay, this might be a dumb question.
No dumb questions.
How does it keep from killing red blood cells if they don't present MHC?
They're trained to look like puppies. These are puppies and we don't kill puppies, okay? Boing, boing, boing. Little red blood cells. So they're just trained to know what self is, okay? Does that make sense? We don't kill puppies. Who kills puppies? Okay, so, the other thing that we're going to talk about, and again, this is a different form, this is an NK cell, we're talking about this now. So we're going to have killing of cells, but the other thing that NK cells do is we have a feedback loop between NK cells and macrophages, okay? So with NK cells and macrophages, so macrophages, when they get activated, one of the interleukins that we saw when macrophages get activated was at IL-12. IL-12 activates NK cells to be killier. They even want to kill more. Okay? So, IL-12 activates NK cells. NK cells then go back and kill, and these are NK cell receptors. So again, these are the receptors that are looking for the activation signal, right? So, NK cells are going to find and activate macrophages by producing something called interferon gamma. So, we're going to find out that interferon gamma is one of the most important cytokines in the immune response. Therefore, IL-12 is also one of the most important cytokines in the immune response because these drive the productions of each other. It's a feedback loop. So once the macrophage starts getting interferon gamma, it's going to make more IL-12. Okay, does that make sense? Okay. For today, we're going to finish up with, so these are the innate, the book talks, they don't give you any images. So these are the things that I just wanted to show you from the book because there's no figure to go over it with. So we're not going to discuss NKT cells in any depth. We're not going to discuss marginal zone B cells in any depth. But I am going to mention, we mentioned gamma delta T cells. And gamma delta T cells are the intraepithelial T cells. They don't go through the thymus. They actually recognize a different form of antigen than the ones of the acquired immune response, our alpha betas. We also have B cells, just like we have dumb T cells, T cells with dumbed down receptors. We have these B1 B cells. And these B1 B cells have a reduced B cell receptor. They have kind of a static B cell receptor. We know exactly what kind of B cells, what kind of things they bind to. And then again, they produce a specific isotype, specific flavor, and they only produce IgM. They do not class switch, okay? So these natural antibodies are actually our ABO antibodies that we all have produced in the body that we're going to go over when we get there. So these B1 B cells are the ones that produce the IgM that makes us need our ABO type of blood specifically. And we'll talk about that when we get there. Does that make sense? You're not going to need to know NKT cells. Just know that they are actually, the immune system is even more complicated than what we're going through. Okay, so there's other things that we're not going to get to, just for time. Okay, so here's complement. I'm going to skip over complement. I'm going to talk about inflammation first. Because we're going to talk about inflammation. And then I'm going to assign you complement for tomorrow. So, for complement, I want you to specifically read chapter 2. This is the information on chapter 2 about complement and the complement pathway. Okay? In chapter 8, they have a more detailed complement pathway. And the only reason they talk about them in chapter 8 is because one complement pathway gets activated by the presence of antibodies. Okay? That's the only difference between why they put it in chapter 8 instead of keeping complement. Complement is an innate immune response to bacteria. We'll talk about these tomorrow. I want you to go through the pathways and try to figure out what activates what. I'm going to give you guys a big hint. The only difference between the classical pathway and the alternative pathway, the only difference is what triggers it. Okay? The only difference, if you look at everything past the very top, everything is exactly the same, except what triggers it. The weird one is the alternative pathway. And this is probably the primary one, but the primary one was discovered after we identified the classical one. So it's too late to call this one the classical pathway because we've already named this one the classical pathway. Okay? So this was the first one discovered because they were identifying immunoglobulins. And what did immunoglobulins do? And they found, hey, immunoglobulins can activate complement. Boom, classical pathway. Ha ha ha. Then all of a sudden they went back and looked and it's like, God, fork. We just found out that this is actually the quicker, faster, better one. Okay? And it's got a completely different and nomenclature. Why didn't they complement people? I want to just punch their little noses. Why didn't they keep it simple, similar to, why didn't they just call it C10, C11, and C12? That would have been just as easy. Instead, they throw in these factor B's and factor D's and factor I's and all of these other factors just to confuse the hell out of us. The other problem with complement is the numbering system is slightly off, only in the beginning. Okay? You will see that C1 activates C2 and C4. What's missing? C3 comes in later. Okay? Only because they named them in the manner in which they were identified. First molecule, C1. It binds antibodies because we're looking at immunoglobulins. That's C1, ha ha ha. Then this is C2, and then this is C3, and then this is C4. Oh, fork. C2 and C4 work together. Okay, so just it's a good thing to test people on. So I like to call it the complement people, or the people who give immunologists something to ask you about, to keep it, because if it was C1 activates C2 and C3 and then activates C4 and then C5, it would just be too logical, wouldn't it? They have tried to go back and make amends, and they can't because there's too much literature out there with the nomenclature already established. Complement people. We don't even talk with them. They have their own meetings, right? Okay. So now I just want to briefly go over inflammation. And we're going to go over inflammation because inflammation is the most important thing about how things get around and what they do. Okay? Once we get inflammation, once you have a paper cut, those PRRs, those pathogen recognition receptors, toll-like receptors, activate, cause the production of IL-1, cause NLRP3s, cause an inflammasome, cause pro-IL-1 beta, cause caspase, pro-caspase. They get activated, we make IL-1, we have inflammation. This is the sense of mediators. So these are some of the main sources. Mast cells make histamine and prostaglandins. They are the best source, and the reason they're around circulatory system and capillary systems is because the job of mast cells is to make the capillaries leakier, so the cells of the immune response don't have to be just PMNs. Those big old fat macrophages can get through. They can't get through without some help, some leakiness. So histamine and prostaglandins help make this leakier, okay? We're also going to have PMNs. PMNs are only found in your circulatory system unless there's an inflammatory process going on. They circulate in your blood and you die unless there's an inflammatory response. They circulate in your blood and they die if there's nothing that happens. In order to get into the tissues, they have to be called out of the bloodstream into the tissues. It's like being on 465 in North Carolina around Raleigh. There's something, or it's 440. It's 440 around Raleigh, right? You have the inner and the outer belt line. In Indianapolis, it's 465. There's these loops around these cities. And you can't get into the city without slowing down, branching off, and migrating out off of these main highways, right? So, what we're going to do is we need to be able to pull these things out of the circulatory system where the bacteria, where the pathogens are. And if it's an interstitial bacterial infection, it's going to be not in the bloodstream, it's going to be someplace in the tissues. So, this is what you need to know. And this is how this happens. All of these things, I'm going to see if I can get to this blink animation because I think they took this down. I think they took this site down because it used to be that you have to buy this. And now I have to buy the website from Amazon. So now you can't get it, you have to buy it. That's so sad. We used to be able to see this online. Okay, so here I have another animation from my other textbook. So this is the exact same cartoon, only animated, that is in your book. So notice the first thing, this is the tissues, this is the dermis, this is the epidermis, these are the basal cuboidal cells, right? This is, what is this? PMN, because it's a polymorphonuclear cell. Unfortunately, they use the same cartoon for every PMN. So does it look polymorphonuclear? No. Okay, but these are the PMNs. This is the circulatory system. your skin has not been breached, okay? So I'm going to turn off this because I'm going to talk through it, okay? Your skin has not been breached here. And at this point, you have a breach of your wall. And when the wall is breached, through the wall comes invaders, bacteria specifically. They've gone through the wall, they're in the dermis. They're going to start growing exponentially. They have on their surfaces PAMPs, right? They have flagellin, they have peptidoglycan, they have LPS, they have those things. They're going to activate. What they should have here are macrophages that are responding and making IL-1, okay? We're going to see macrophages come in later, but as soon as those things are there, it should be triggered. The other thing that's going to happen that you guys are going to talk about, we're going to talk about tomorrow, is that as soon as bacteria are here, the alternate complement pathway gets activated. Boom. Okay? So complement molecules are being released. We're going to talk about C3A and C5A are actually breakdown products of complement that drive inflammation, just like IL-1, TNF, and IL-6. IL-1, TNF, IL-6, C3A, C5A. All pro-inflammatory cytokines. But those C3A and C5A are breakdown products of inert molecules. They only get activated in the presence of bacteria. So once those are made, they're going to start activating. These are mast cells. Notice the mast cells are around the circulatory system. So here we have the mast cells around the circulatory system. PMNs are rolling along, and they're too small and they're moving too fast. They're too small and they're moving too fast for them to get out of the bloodstream. There's nothing for them to hang on to. These have to crawl on surfaces. They can't swim. They can't migrate through fluid. They have to crawl on the surface of things, which is why bladder infections are a problem, because if you have bacteria in your urine, they can crawl on the outside of your bladder, but not get the bacteria that are in your urine, right? So PMNs can only crawl along the surfaces. And here they're moving about 450 miles an hour. Boom, 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 boom. Every time the heart beats, boom, 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 okay? So we've got these moving very rapidly, okay? So now IL-1, IL-6, TNF, C3A, C5A are being released. It's going to act on these mast cells to degranulate. And the other thing that happens, we're also going to upregulate something called selectins. And these selectins are named selectins because they are selecting, you can think of it as selecting the tissue where the PMNs can exit from. If you have a cut in your arm, do you want them to exit in your leg or on your butt or on your arm? You want them in this part of your arm where the inflammation is. So these selectins are only upregulated where these inflammatory signals are being generated. Inflammation is generally local, okay? Fever is whole body, inflammation is local. We talk about redness, where we're going to have more blood cells coming in. We have more blood coming to the site. Swelling, all of the fluid, all of the plasma is now going to become interstitial fluid. And it's going to pull in complement proteins and antibodies along with it, okay? Heat and pain, heat comes from the PMNs migrating in because they're going to be enzymatically active. And pain is because the swelling causes the nerves in that area to swell. So we have redness, swelling, heat, and pain occurring in this localized area. Because when you have a paper cut here, are you having any sort of response here? You're not. It's localized. So we're going to have this. And so these slow the PMNs down. This is called marginization. And so these mast cells are now slowing down. So now, once they start slowing down, on the surface of the PMNs, and you'll see
adhesion receptor molecule.
On the surface of the PMNs, there are selectin receptors. And we just call them, in the book they might talk about them more specifically. I know I'm a little late. These are selectin receptors on the PMNs. Once the selectin receptors start binding to chemokines, attractants, cytokines, they're going to upregulate, they're going to change the conformation of this molecule that's on the PMN called an integrin. Selectins allow the PMNs to slowly roll along. Integrins allow them to stop. And so there's an integrin receptor. Selectins are on the epithelium, Selectin receptors are on the PMNs. Integrins are on the PMNs. Integrin receptors are on the epithelium. So think of it as Velcro. There's always one side. Both of these things, Velcro slows, there's one that slows them down, there's the other one that allows them to stop. Okay? So, they're going to slow down. The integrins then bind to the integrin receptor. They interact. The mast cells are going to degranulate, cause histamine and prostaglandins to be released. Is it still going? Oh no. Did I stop it? It should be still going. Okay. Oh, it's showing you that there are more and more bacteria. Histamine is released, and histamine causes vasodilation, which allows these capillaries to become leaky. Plasma is going to start leaking out. Complement proteins, antibodies that are in the plasma. Because remember, antibodies are only about that big, right? They're going to be leaking into the tissue. PMNs migrate into the tissue. They bind to certain complement molecules. Extravasion, they migrate in through, or it's also called diapedesis. And now they're phagocytosing those and using phagocyte oxidase and nitric oxide synthesis, inducible nitric oxide synthesis to create molecules that allow them to destroy those. Okay? Does that make sense? And that's that. So this is exactly, it looks like the same thing. There's lots of inflammation animations, but that's just as good, that's what you need. And so we're going to talk about complement tomorrow and finish up the innate immune response tomorrow and start on chapter 3. Okay? And these are just the last, yeah. Okay. Go over complement pathways. Okay? Just a minute, let me turn this off.